I'm delighted to introduce a colleague, Anne Young, who's been working in biomaterials and tissue engineering at the Eastman Dental Institute for a number of years and has a major interest in tooth filling materials. And she's going to be talking today about that and also about the process and journey that she's been on moving these towards clinical trials and CE marking. Thanks very much, Anne.
Okay, thanks, Jonathan. Thanks. So today what I'm going to do is talk about our work on the development of composites for painless restoration of children's teeth. As Jonathan has just said, we're trying to optimize the formulations, get them manufactured, and then into clinical trials, and ultimately we're looking to get them CE marked. So what we want to have is what we call at the moment a smart, strong dental composite. What we want is to be able to place without having to use any drill or anaesthetic directly on minimally excavated children's teeth. What we need is for this smart composite to have multiple unique selling points. In particular, we want it to be able to be self-sealing. What this can do is it's self-sealing. It will prevent both the bacterial and the enzymatic action that cause degradation of the tooth structure. Also, to have a material that is going to last a long term, we want the material to repair itself and to remineralize the tooth surface. We're aiming to have something that can treat what we consider at the moment to be almost unrestorable teeth in less than 5 minutes. What I plan to cover is, first of all, the causes of decay, its current restorative chemistry and the different problems we've got, and then go on to smart material properties, what you need to do if you want to manufacture in order to undertake clinical trials, and also some of the processes that you need to go through in order to gain CE mark, which is what you need to get to market. So dental caries, it's one of the most prevalent diseases worldwide. It affects 40 to 60% of children and 90% of adults. In the UK, it accounts for around about 3% of all the NHS healthcare costs, and globally it accounts for about 5 to 10% of all healthcare expenditure. It's caused by bacteria, specific bacteria like strep mutans, Sorbrinus, which converts sucrose from food into organic acids. What these do is they chelate the calcium and dissolve the phosphate and hydroxyl groups that are in the apatite that's in both the surface enamel and the underlying dentine. What then happens is once the dentine is demineralised, you get enzymes being activated that hydrolyse the remaining collagen. If we look at a carious tooth section, then what you can see is that the top dentine surface, that's highly degraded, highly infected, soft, and easy to remove, but it's beyond repair. If we look underneath that, the dentine is demineralised, but the tubule structure is often still intact, and this dentine is actually repairable. So what we're looking to do is have effective sealing that starves the bacteria. So if we don't have any more food getting down, then the bacteria can die off. The materials we've got for tooth restoration include in the EU the amalgams. So these have been available for nearly 200 years. They consist largely of mercury, around about 50%, that's mixed with other metals, silver, tin, copper. What happens is you generate a paste, but in order to place this, you need to make an undercut in the tooth. The big advantage is that the materials, they are strong and they're antibacterial. And they've got a good lifetime of around 9 years. But in 2018, the use of amalgam was banned for children in the EU. And this is because of a Minamata agreement to phase out globally all use of mercury. The alternative that's been available since around the 1960s is the bonded composite. This consists of around about 80% of very fine glass particles that are dispersed in polymerizable liquid methacrylates. What you get is a paste that when you expose it to blue light, it sets solid. It's much preferred by patients because of its aesthetic appearance, and it's bonded to the tooth. Because you get bonding, it's more conservative of the tooth structure, but the bonding procedure is really complex. The composite is not antibacterial, but what it does is it seals in the bacteria. The issues are that with a composite, median lifetime is only around 6 years compared with about 9 for the amalgam restoration. And the failure is usually because of debonding and recurrent caries at the tooth restoration interface. It's the bonding complexity that makes the composite really far from ideal for treatment of very young children. You need to have an anaesthetic, a drill, tooth isolation and a rubber dam in order to be able to place the phosphoric acid etchant. Then you have to rinse and dry. Following that, you add multiple steps of primer, adhesive application, drying, curing. And then you have to place the composite in thin, usually 1 millimeter thick layers, and cure each layer in turn by exposure to a blue light. Once you've drilled the tooth structure, what you have left on the surface is what's called a smear layer that's contaminated with bacteria. If you treat that with the phosphoric acid gel, that will kill the bacteria, but it also etches the enamel, producing a rough surface that is relatively easy to bond to. What you also do, though, is you etch the dentine structure. What this gives you on the top surface of the dentine is a layer of around 2 to 4 microns thick, which is made up of collagen fibrils that are demineralised on top of the sound dentine. What it also does is it opens up the tubules, the dentine tubules. In order to get bonding to this is much more difficult than to the enamel because these tubules contain water and they repel the hydrophobic composite. So what you need to do is have a primer that can penetrate deep down into these tubule structures and also penetrate into the collagen fibrils. What you form when you polymerize the primer is tags of polymerized resin that seal up the tubules, and on top of that a hybrid layer. If we look at the composition of the dentine primer, it usually consists of HEMA, which is hydroxyethyl methacrylate. What you have in HEMA is a hydrophilic OH group, which allows it to dissolve in water and mixtures of water and ethanol. But it also has a methacrylate group, a C double bond C group, that is polymerizable. Additionally, you often find acidic monomers in the primers. What these have is the methacrylate group, as in HEMA, but often a phosphate group or a carboxylic acid group that combined with calcium in the apatite, providing a bond between the resin phase and the remaining apatite. After applying the primer, you have the adhesive. The adhesive often also contains HEMA, but what we do is to replace the aqueous phase, the water and the ethanol, with components that you would find in the dental composite. This includes components like bis-GMA, which is a large dimethacrylate monomer, and varying levels of different filler particles. The problems, though, HEMA, it's a small molecule, it's cytotoxic, it has high shrinkage when it polymerizes. You can also have the hydrophilic and the hydrophobic components phase separating from each other when you try and mix them. And you can get pockets of water where you can get continuing bacterial action, continuing enzyme action, causing degradation of remaining collagen and dissolution of apatite. Also, because the hema, when it's polymerized, it's still very hydrophilic, so you can draw in water, and that can cause it to degrade. The alternative for children is the glass ionomer cement. Glass ionomer cement, this is composed often of a 50% polyacrylic acid solution, aqueous solution. So polyacrylic acid, it has a carboxylic acid group that is reactive with both the tooth structure and also a glass that is combined with the cement. The major advantages: you can place material in a single step, you can use a minimal excavation method, only taking out the top surface tooth structure. You usually don't need to condition the tooth. Instead of with phosphoric acid, you use a solution of the polyacrylic acid. The benefit as well is that you can get release of fluoride. What the fluoride can do is help to get the tooth underneath the restoration to remineralize. Disadvantages of the GICs: they have a slow setting reaction that is moisture sensitive. But the biggest problem is they tend to be weak and you can get fracture of the restoration. So usually glass ionomer cements are considered as a temporary or a base restoration material, so they can be placed underneath, for example, another composite. What companies have been trying to do is produce a wide range of different hybrids. So some of the early hybrids were the acid-modified composites or the resin-modified glass ionomers. So the acid-modified composites, those are strong and they provide some fluoride to try and encourage remineralization, but you still have to have the prime and bond steps to get the material to stay placed. The resin-modified glass ionomers, their benefits: they do bond like a GIC, they do release the fluoride, they have much better strength than the conventional glass ionomer, but they contain hema. The resin is hema, which is cytotoxic. Other companies are producing flowable composites. To produce a flowable composite, what you do is reduce the amount of filler. This helps the material to adapt and bond to the tooth, but you can get then a higher shrinkage with the lower filler. You tend to also often get lower mechanical properties. What some newer materials that are coming online are things like Halt Dent's Activa. What Halt Dent have done is they produced a material that they call being a bioactive material. What this does is it releases calcium, phosphate and fluoride ions, which can help to remineralize. But it's still, this material really still needs a prime and bonding agent. The biggest problem, though, is for the very young children. What we've still got in the UK is 25% of 5-year-old children having caries. And at the moment, less than 10% of those children are being treated in our general dental practice. What's happened over about the past 10 years is, because of phasing out of amalgam and many other issues, the numbers of children being referred to the Eastman, where I work, has gone from around about 2,000 to well over 5,000 being referred each year. As a result, we're getting increasing waiting lists. In the UK, waiting lists for children before COVID were about 4 to 5 months. In some hospitals up in the north of the UK, that can extend to a year. With COVID, we've not been treating now for a further 4 months, so those waiting lists are going to be much, much longer. And there are other issues now in trying to place a composite material. It generates aerosols which can spread the virus that causes the COVID-19 pandemic. The problem for the children, they've got these very long delays. During those delays, they have risk of sepsis. They're having repeated bouts of pain, sleepless nights. And also when they do finally get to be seen, they can have more extensive decay. And for many of them, the only option by that stage is to have the tooth extracted. This is costly for the NHS. It can also cause issues such as bone loss, tooth misalignments, and other issues such as dental phobia. So what we're looking to do, produce something that can seal, that can be placed directly on this highly carious dentine. We're looking to be able to do what we can do with a GIC, but with a large cavity rather than the small cavities. So we want the sealing to stop both the bacterial and the enzyme action, and we want treatment in less than a minute, less than 5 minutes. We also want the material to repair itself and to remineralize the surface of the tooth structure. If we look at a conventional composite, it consists of around about 20% of dimethacrylate monomers with an initiator and an activator in there. The monomer is usually bis-GM. Problem with bis-GM is it's got a couple of benzene rings in the middle of the structure. That makes it a very rigid molecule, and that can slow down its ability to polymerize. To solve that issue, what we've been doing is removing the bis-GM and replacing it with a much more flexible molecule. What this allows us to have is much faster polymerization and more extensive polymerization. It also means that we can take out the activator that was in the conventional composite, which is known to be cytotoxic. When you expose the methacrylate to light, what happens is you start a chain reaction that joins all the methacrylate monomers together. What we found is we can use a Raman technique in order to look at how much monomer is remaining. What we find is that there is a peak that's due to vibration of this carbon-carbon double bond, that when the material polymerizes, that peak is lost. So the height of the peak after polymerization, divide that by the height of the peak before, tells you how much monomer is left in the system. What we're also doing is developing an infrared technique that allows us to quantify how fast the process goes. What people often do with infrared is use a peak that's due to the C double bond C in the infrared spectrum, but that's not useful to use if you've got water present because the peaks are overlapping. So what we're using is a different peak that's due to vibration of a carbon-oxygen bond. What happens is when the monomer polymerizes, that peak shifts. And so again, we can use the height of this peak to see how quickly the reaction, how quickly the process is going. What these results are showing is that when we turn the light on, we get very fast reaction, very fast loss of very fast loss of the methacrylate peaks. However, when we turn the light off, the rate of the reaction slows down. With infrared, what it's also possible to do is look at what happens if you have samples of different thickness. We can look at what's happening at the bottom of samples of 1, 2, or 3 millimeters depth. What we've found is with the smart composite, if we have a 1 millimeter sample, there is a 2-second delay before there's any reaction. If we double the thickness, there's a 6-second delay. And if we go to 3 millimeters, there's a 12-second delay before there is any reaction. What this means clinically is that if you have a thin restoration, you only need to expose to light for 20 seconds. But if you have 3 millimeters, 2 millimeters, you need to apply the light for 40 seconds to get our maximum conversion. The other components that we've added in, first of all, we've added in an adhesive monomer. So this is the monomer that we find in the primers, and that helps us to get some bonding to the tooth structure. We've also added in 2 other components that we find in food. First is polylysine, and the second is monocalcium phosphate. Now these we've actually only had to add in low levels, but they make a really big difference in the way that the material interacts with dentine. So polylysine, our first additional component from food, it's got FDA approval as a preservative for food. And it's also got what's called BRAS status, that's generally recognised as safe. The reason for this is it is highly compatible with our eukaryotic cells. In fact, it's actually used in cell culture to get cells to grow on the surfaces of plastic. We can also find that it's added into a wide range of different foods, with rice being particularly common. It's added in at levels of around about 0.1 grams per kilogram. But in the body, what it does is it breaks down into the essential amino acid lysine that we need in order to produce more collagen. In the daily intake of lysine can be anything up to 4 grams a day. In Composite, what we've found is that the polylysine absorbs water out of the tubule structures, allowing the resin to penetrate in. So what this does is it seals off the tubules, stops nutrient ingress, which would stop the bacterial growth. We also stop the enzyme action, which causes the collagen degradation. In order to look at how different components affect the sealing ability, what we've done is we've had to make a disease-affected dentine model. To do this, we've taken 2 millimeter thick coronal dentine slices, and we've combined them with formic acid solutions, 4 molar formic acid for 48 hours. What that does is it dissolves away all the calcium and all the phosphate, all the mineral, but it leaves the collagen structure and the collagen tubules intact. We can then place the resultant flexible disc of collagen that we've got left, place that on top of different composite pastes. We then polymerize the paste and dissolve away any of the remaining collagen with sodium hypochlorite. And what we get left with is, on here, this is the composite. We've got an imprint of the collagen, the dentine slice. And you can see dark areas here. And in these dark areas, what we find when we look under SEM are really long resin tags. From elemental analysis, that's EDAX, we can see that these tags are composed largely of the resin components. They're not the solid, they're not the polylysine, it's the monomers that are forming these tags. If we take this dentine that's demineralised, place it on top of a commercial material, the two surfaces are repelled by each other. The composite is hydrophobic, it doesn't like the water in the dentine. Experimental smart composites, what these do is they penetrate in. If we want to get very long resin tags, we need to add in more monomer into the system. If we're at about 20% monomer, then tags are only about 10 microns. If we go up to 25%, we can get tags that are 200, even more, in length on the surface of the dental composite. But what determines what we call the coverage is the powder content of the composite. That's the hydrophilic polylysine monocalcium phosphate that we've added in. What we find is if we have low levels of those two components, the tags are very patchy. We have some areas that have got tags, but other areas here, these have got no tags visible. What this has done is helped us to optimize the formulation, to come up with a formulation that seals practically the whole surface of any dentine tubules. What these results here show is if we take that optimized formulation and take out our polylysine, our coverage goes down from around 85% down to just 25%. Polylysine is the most important. If we look at polylysine particles, what they are, they're about 50 micron in size. They're far too big to penetrate down into the dentine tubules, which is why we believe that what they're doing is they're drawing out the water from the tubules to allow the resin to penetrate. What we can see here, the calcium phosphate and the adhesive monomer, they do have some benefit, but the benefit is less than the benefit of the polylysine. Additionally, what we've done is we've used this dentine model to look at how enzymes are working on the surface, on the interface between the demineralized dentine and the composite. So we've taken demineralized dentine, we've added a probe that fluoresces green when the enzymes are being active. We've then restored the top surface, left it incubating for either a day or for 14 days, and then sliced through the restoration to have a look at that interface, to look at those enzymes, see if they're actually active. And what we find with all these different commercial materials, Z250 is a composite, Fuji 9 is a GIC, Fuji 2RC, a resin modified GIC. And this is our smart composite with the polylysine removed and smart composite. All of the commercial materials, they've got extensive green fluorescence showing that the enzymes are active at the restoration interface. Smart composite, there is some activity to start with, but 14 days is nothing. So they're really managing to stop this enzyme action that would cause degradation of the collagen. So we've then gone and proved that the same thing happens if we take a highly carious tooth. We've restored a highly carious tooth that's been extracted. And again, we get the same effect. Commercial materials, the enzymes are active at that interface, Caries interface. So the other component that we've added in that's from food is calcium phosphate. It's used as a source for calcium phosphate, which we need for bone growth. But it's also used in food, in baking, to make bread rise as a leavening agent. In the composites, as we've shown, it promotes, helps to get us having this tag formation and sealing of the restoration interface. But what we've also seen, if we take a composite and fracture it and then leave it in the simulated body fluids, the cracks actually begin to fill with a calcium phosphate that seals up any of the fracture sites. So it helps us to get a self-sealing, a self-repair mechanism. But additionally, if we place our material in contact with demineralized dentine and then take it away, what we see over time is that we have mineral precipitated at that surface. If we do this with commercial materials like Adhesive, Fuji 2 that release the fluoride, we don't see anything at the surface. So what we've done is we've placed this polymerized materials on top in contact with the demineralized dentine. Smart material, it gives this layer of calcium phosphate after 6 months, which can help us to seal up any gaps that might form over time. One of the issues is that if we have very hydrophilic components, they draw in water, and that can cause strengths to go down. If we look at a commercial material, they all go down in strength somewhat in about the first 28 days after placement in water, due to water absorption. And the materials we've got, they decrease in strength by similar amounts over 28 days, but then form an equilibrium strength. If we compare smart final formulation strengths with those of commercial materials, here we've got Activa, the new bioactive composite, a resin modified GIC and a GIC. It's much stronger than the conventional GIC. It's above the 100 mark that's required for a composite, comparable with Activa, stronger than a resin modified GIC. So it's a reasonable strength. It's strong enough, particularly for restoration of children's teeth. So having optimized the material, what we're looking to do at the moment is full-scale manufacture. And to do this, we've been working with companies that have ISO 13485. This is a certificate that's required in order to be able to manufacture. What DMG Schottlander have done together is they've produced a number of different formulations, batches, proved that they can make exactly the same as what we can make. They've gone from 10-gram batches up to 2-kilogram batches, and they're starting now to find different ways to scale up filling of compules. And what Schottlander have done is they've registered a trademark for this new smart composite. They're calling it Renewal MI. We've also had to do a number of cytocompatibility testings. We've done a lot of this in-house, but we wanted to confirm that the results we get can also be obtained in laboratories that have GLP, good laboratory practice. They've looked at properties such as cytotoxicity, carcinogenicity, and others. So once we've managed to manufacture, what we were able to do is to apply to undertake a clinical trial. If you have a product that doesn't have a CE mark, you need to apply for both ethical approval and regulatory MHRA approval in order to undertake a clinical trial. What the regulatory authority, the MHRA, want is information summary of all of our bench testing data, all of our biological assessments. They want an investigator's brochure that explains how the material works, instructions for use for the clinicians, and evidence that we comply with what's called the Medical Device Directive. In addition, we had to produce a clinical investigation plan. So what this plan was is to place material in the teeth of 6 children who had teeth that needed to be extracted because they were so badly decayed. But what this meant was, because the teeth were being extracted, we could place the material, and then when the tooth was extracted, we could look at the restoration material in a lab. We also needed to have patient information sheets, consent forms for both the child and for the parents. What we managed to show in the clinical trial is that the materials could be placed in minimally excavated teeth. The procedure took less than 5 minutes. There were no adverse events. The materials stayed in place for the full month. Children had no pain during that month. And when we looked at the materials in the lab, we found that if the restoration had been quite small, then when we dyed the material, the restoration, and looked at the interface, we could see these long resin tags that we had seen in our laboratory studies during the development of the material. When material was placed up into some really large cavities, what we found was that the composite completely surrounded all the infected dentine, interacted with it, bonded it, sealed it up, and again stopped the child suffering from pain. So having undertaken a first clinical trial, what we're now looking to do is try and pull together all the information that you need to get to CE marking. In a technical file for CE marking, what we've got is an archive file, a background file, and a cardinal file. So the archive has all of our information on our granted and licensed patents, our grant applications, all the many reports we've had from all the different funding sources. We've had a lot of students working on the development of these materials, doing lots of different aspects of how materials interact with teeth and optimizing the formulations. What we've also done is have the background file that contains a lot of publication and technical reports that have been produced by postdoctoral scientists, looking at what are the components. You have to do is specify what all the components are, where you're going to get them from, what are their purities, and all that kind of information. Then in our cardinal file, we have all of our data, all of our final formulation data, in vitro testing summaries, clinical trial documents, how we conform with the medical device directive, all of our biological safety assessments. So where we're at now, we're looking next. If we get the CE mark, we're wanting to go for a 60-patient clinical trial for 6 to 18 months. If we don't get the CE mark, then what we need to do is we'll have to do the patient trial before getting the CE mark. But to do that without the CE mark, we've got to go through the regulatory authorities again. What we'll be looking to do is see that we can get a failure rate significantly below that of a glass ionomer cement. We're also doing some work with Thailand. In Thailand, they've got a much bigger problem with caries in young children, whereas we have a level of around 25% of 5-year-olds. In their rural areas, it can be up to 75% of their 5-year-old children have got caries. So they're particularly interested in a material that is very easy to place. So our main aim is then to see if we can get this material used and trialed in general dental practice, to see if we can try and overcome the problems of treating nervous patients. So in conclusion, what we've got is a smart composite that can be placed in a single step, sets and seals the dentine in less than a minute. It has reasonable mechanical properties. It's able to prevent the bacterial and enzyme action through sealing, and it's got unique remineralising, self-repairing features. So hopefully this will allow treatment in less than 5 minutes. It remains for me to acknowledge the vast numbers of students and staff that have done all the work that's been described. We've also had a lot of funding from research councils, from the NIHR, and also from industry, from Schottlander and from DMG. And thank you all for listening today. Thank you.